Hello, and welcome to the new lecture on studying the circuitry of the brain. Um, so, what exactly is the brain, right? So, we know it is inside our heads and is involved in controlling most of our functions. The brain itself is made of neurons, which are specialized eukaryotic cells or animal cells. And we have approximately about 86 billion neurons with all their connections. And this results in a phenomenal machine, if you want to call it that, a processor with very little power usage, right? So even if we build a machine that is analogous or equivalent to a brain, we always end up with the significant problem of how to power this machine, right? That's the main reason why we want to study the structure. And in this talk, we will talk about some of the ways as to how we can actually do it. Okay. To give you an idea about the scale of the problem that we encounter when studying this structure, if you actually map out the connections between individual neurons and if they were your noodles or spaghetti, um, they will end up occupying the entire volume of Mount Everest, which is the largest mountain structure on the planet, right? So that's how complex the issue is, and we want one of our goals is to understand what this circuitry is and how we can entangle them, if that's the right word to use. Okay, the second part of it is why exactly do we want to study our brains, right? Let's assume that it's a machine, a pretty smart machine, and the closest we have to a proper brain in terms of a machine is IBM Watson, which was built, it showed its prowess on a quiz show. But IBM Watson had a significant drawback, which is the power usage, right? The Watson actually had 90 Power 750 server machines, which was taking, two times 1,925 watts of power. Right? To give you an idea, this works out to be approximately the power substations for about 2 suburbs. That's a lot of power that it uses, right? All this for beating somebody in a quiz show, right? Your brain does a lot more than that, right? Not just remembering things for a quiz show, you also do all your daily functions and everything for that. But how much power do we use? The power do we use is about 20 watts, and that's equivalent to your light bulb, right? So that's another reason. So we need to understand how does the brain actually do it, and then can we implement it back into a machine? So let's go to how exactly do we do this, right? So this is actually called neuroanatomy, and basically neuroanatomy is defined as the study of structure and organization of the nervous system. Now, this is very different to neurophysiology, which is basically finding out what is the function of a particular brain area and then figuring out what is happening. The main fundamental things that we study in neuroanatomy is the neurons themselves. We'll call it, sometimes people call it cells. And in a machine term, we will call it nodes. And study of this is, otherwise sometimes we use the word cytoarchitecture. The second thing that we want to study is what are the different connections between these nodes. Okay, so these are sometimes referred to as fibers or connections, and we will also intermingle with the use of the word myeloarchitecture here. The third thing you want to study is areas of significance. For example, visual cortex has 40 different areas, each with its own different functions. So let's say a primary visual cortex or a face area. What exactly does it mean? Okay. The fourth we want to study is where exactly do these neurons come from? After all, I just told you they are all animal cells. They're specialized, but they're still animal cells, right? So they all come from the, if I want to call it, the same stem cell or origins of it. But what makes them special? What makes a neuron and not a hair cell or a skin cell, right? Now this is a specialized area. It's called genomics, and it'll be covered in a later lecture. All right. The next part that we want to encounter is the pathology. Like how can a brain get infected by bacteria, viruses, concussions, accidents, what causes it to, these kind of things. It's quite relevant at this day and age, simply because you have the pandemic spreading across the world now, and COVID-19 certainly does affect the brain. Patients have been complaining about what is called as brain fog. We are still yet to understand why exactly it happens. Now, at what scale do you want to study the brain? Okay, the brain is a fairly, the human brain particularly, is a fairly large biological tissue, approximately 15 centimeter by 15 centimeter by 15 centimeter. Now, there are several ways of studying it. What you see here is what we define as a macro scale. Basically, that's what you see on the left-hand side is a whole body scanner. So, obviously at the scale that we're studying, that's about 2 meters approximately, which is the height of a human being. And a part of it can be considered as as I just mentioned, a 20 centimeter scale. This is probably what you do in your MRIs, right? So you go into you had some issue, you want to scan what happens in your brain. It's a gross structure, you want to have a look at it, and the way you do it is you do a magnetic resonance image. So that the what you see on the right hand side is actually a MRI. Okay, so this scale is great if you want to look at very gross structural abnormalities, but it doesn't really give you a picture of what is the neuron, where are the connections, and so on and so forth, right? This is the next scale that we want to do, right? Which is basically, this is histology, and what you see there on the left hand side is actually a mouse brain. Mouse brain is approximately 1 centimeter by 1 centimeter by 1 centimeter, and so this particular tissue has been stained for a dye that is very specific to neurons. It's called Nissl staining, and the chemical that we use here is cyanin. And you kind of notice that there are different structures here. There's different patterning of these cells, and I can zoom into the scale and look at some of these neurons. Okay. Now we're going even further. We go into what we call as electron microscopy / microscopic scale. And here we can actually identify all the structures that are inherently present in a neuron. So the scale that you see on the left-hand side is about 500 nanometers, which is half a micron. And the scale that you see on the right-hand side is about 20 micron. There you can see the entire neuron. It's been stained with special fluorescent stain, so you can actually see the cell body and the associated axons and dendrites. Now, which of these scales? How do I know what to choose? I have all these tools at my disposal. So let's make a decision as to what kind of technique that we want to use. What are the advantages and disadvantages of this technique? In addition to the scales, there is a second component, which is it depends on depending on what species you want to study. The brain size actually varies, right? So the human brain, as I just mentioned, is fairly large. So it's about 15 centimeter by 15 centimeter by 15 centimeter, compared to, let's say, a mouse, which is 1 centimeter by 1 centimeter by 1 centimeter. And this is particularly important, right? Because obviously you're going to be spending a lot more resource allocated to studying a human brain compared to a mouse brain. And the overall size is also gets extremely important when you're studying neuroanatomy. So the choice of your technique and scale depends on how big or how small is your and what you're studying. Right? Let's go to the finest scale possible, which is the electron microscopic scale, right? There are two issues. We'll start with the basic problem, which is there are two issues here. One is It's a significant data and a time problem. If I were to image a human brain, on all its glory at an electron microscopic level, at around 50, 50 nanometers or so, um, the data set that I'm going to generate is approximately 175 exabytes, right? For one human brain, let's, for n equals 1, that's 175 exabytes, right? To give you a perspective of what an exabyte is, approximately 300 exabytes is the entire storage capacity of the planet, right? All the storage, including your laptops, my laptops, everything taken together is about 300 exabytes, which basically means, I can't even, study two human brains because there is not enough data that I can store. There is not enough space to store. That's your first problem, which is the size of the data is massive. The second problem you're going to end up is that it takes a long time. Electron microscopy is a reasonably time-consuming process. And to give you an idea, if you studied 1 millimeter cube, so that's 1 millimeter by 1 millimeter, 1 millimeter, compared with the mouse brain, which is 1 centimeter by 1 centimeter by 1 centimeter, at electron microscopic resolution, at the resolution that we just talked about, 50 nanometers, this will take approximately 1.5 years to collect. And now expand this to 15 centimeter by 15 centimeter by 15 centimeter, we simply don't have the time to collect all the data, right? There is a second problem. The second problem is when you zoom in at that level, at nanometer scales, the majority of, there is some variability, but the majority of structures look exactly the same, right? So, if you were to assume, let's say, the sodium potassium pump, it's made of chemicals, and the chemicals are arranged in a certain format, and this format doesn't change a lot. There is no it doesn't look like there is big variability at that scale. You got it? So which is not the case, right? So your brain is different to my brain, or somebody else's brain, or a mouse brain is different to a human brain. But at that electron microscopic scale, the ability to appreciate these differences is extremely negligible. Okay. And we just mentioned about the other scale, other type, which is your MRI scale. It's great for structure, to look at overall big picture, see if something has gone wrong or something has not gone wrong. But it's not great to look at, you can't even see single cells. Let's make it simple, right? So what do we do? So we'll go to intermediate point in physics. It's actually called the mesoscopic resolution. So this problem has already been solved in physics, particularly astronomy. Our neurons are approximately, if you want to call it, the soma of a neuron is probably approximately between 5 and 10 microns. And then there is an axon, which is pretty long. For example, the axon that supplies your little toe on your foot is about 2 meters long, your entire height when it goes from the brain all the way down, right? So we are creating a compromise, right? So we are going for a compromise where the resolution of 0.2 to 1 micron is actually pretty good to measure something that's 10 micron. Yes, so that's 20 times or 20 pixels or 10 pixels thing, right? Yeah, can we do it? Is the next question. Do we have the capacity or the compute power to actually study and digitize this? Absolutely yes. Is there actually any resources that are already available? Very true. Particularly for the mouse, there is a lot of databases that are available. One particular one is is part of our group, which is Professor Pathamitra. He has the mouse brain, as well as other species brain that has actually been histologically processed and imaged at a reasonable resolution, and it's freely available to everybody, right? The website that you go to look at Professor Mitra's lab data is called brainarchitecture.org, and there are different species brains that are available, including mouse, rat, turtles, fox, cats, right? And to look at, let's say, the mouse brain, right? So their scanner does it at about half a micron per pixel, and one mouse brain ends up with about 101.5 terabytes of data. Okay, and the brain architecture project has somewhere close to two, probably higher than two right now, because the data is obviously is appended as time goes. We have close to 2 petabytes or even greater petabytes of data that is all publicly accessible. So anybody who's interested, please visit the webpage. Right. So okay, now we have established this, right? So mesoscopic resolution is probably is okay, but then comes the next question. Okay, we do this for a mouse brain, it is 1 centimeter by 1 centimeter, or let's say a cat brain, which is a 4 centimeter by 4 centimeter by 4 centimeter approximately. Can you actually do this for a human brain to study at that resolution, half a micron per pixel resolution? Of course, the first problem we are going to have is that is a 15 by 15 by 15 approximately, which is a significantly larger brain. Most of the commercial equipment that is used for histology is designed for biopsies, which means they're very small, right? So we don't even have a proper cryotome or a sectioning equipment. The glass slides that you generally see in a microscope are 1 inch by 3 inch slides. A human brain will not, a human brain section will not fit in there, right? So which means you need specialized glass slides, and yeah, so it will fit in a 6 inch by 8 inch glass slide, just about. The third part we have is, if you want to digitize all of this, we don't have a scanner that can scan 6 inch by 8 inch glass slide. We have one commercial equipment that is available, but it's extremely slow. At full resolution, it takes about 2 hours a slide. And the human brain, if you take 20 micron as a thickness that you want to cut per section, we'll end up with something like 7,500 sections. And if I spend 2 hours per slide, that's a long time to scan just one human brain. The benefit we have in studying at a mesoscopic scale, which is like half a micron instead of the electron microscopic scale, which is 50 nanometers, is that one human brain data set will be approximately 1 petabyte, which is still pretty large, but we have the capability of storing 1 petabyte right now, right? Which is reasonably okay compared with 185 exabytes of data in EM resolution. What does neuroanatomy exactly study, right? So we kind of established, okay, yes, we have some technology that's available. It's probably at the edge. We are at the edge of the technological advancements to study the human brain. But what exactly can you study in a human brain, right? So let's talk a little bit more about the methods right now in the next section. So we're going to be studying the structural architecture. What exactly does mean? Okay, it's basically, if first time you saw the Taj Mahal or the Eiffel Tower, you just went, wow, this looks awesome, right? How on earth did they build it? Right? How does it work? Why does the Taj Mahal have? How did they manage to get a dome on top of the Taj Mahal made of marble? It's too heavy. Okay. So the structural architecture kind of gives that architecture, right? You want to know, what kind of bricks they used, what kind of, how did they do it? Did they do it with cement, or in the olden days, people used to use interlocking bricks? Is that how you use it? So that's what we're going to study in terms of structural architecture. So we're going to be looking at the cells that make up these building blocks, right? The second thing you want to do is connectivity. Okay, how does the visual cortex connect to the auditory cortex, or how does our decision-making center, the prefrontal cortex, connect to the visual cortex for us to make any sort of judgment and response, or to the motor cortex? Something has to make a decision and make a movement, right? How is it actually connected? Right, and the third thing we want to know is, what are the different types of cells that are there in the brain? Can we actually separate them out, and what is the functional or structural significance of these types of cells? How do you do it? You look at, I don't know how many people have seen this, but if you look at a piece of brain, you made a small 20 micron sections, you don't see anything significant. It just looks like a brain. There are, very gross, it's mushy, and you see probably separate two different structures, white matter and gray matter, which we talked about in our introductory lectures, right? But how do you systematically study that? The way we do it is we use procedures that have been well established in neurohistology. These are neurohistological procedures, and the diagrams that you see on the background there are from Ramon and Cajal, who actually pioneered some of these techniques and meticulously hand drew majority of the cells which we are seeing right now, and he's pretty good in what he did, right? Okay, and so how do we do it, right? So I just told you the brain is this mushy structure. It's more like a jelly rather than the mental picture that you get is like a hard structure that's there. It's not hard; it's very soft, but we need to make it hard in order to cut it, right? And the way we do it is we use a procedure called fixation, which basically means now you embed this brain into certain compounds that can bind and make it a little harder. Okay, you would have heard the word formalin before, or formaldehyde before. This is one of the chemicals that is used for this procedure fixation. And now obviously after fixation, we can section it, which basically means you're cutting very thin slices across the brain. And then we'll mount it, on glass slides, and we stain it. Which staining means, obviously we cannot. It's very difficult to see cells directly, so we will use these special dyes which can stain a particular type of cells or particular connections, and that gives you, a good picture of what to view. Um, there is an important caveat that you need to remember in staining, right? You don't want a stain to be staining everything. Okay, once you stain everything, you can't see anything. There is no contrast, right? Everything is overlapping. There's nothing you can see, and you don't want the stain to be so selective that you can't see anything, right? So it's a compromise between whether you can stain your objects of interest alone and sparse enough to visualize it, not too sparse enough that you can't see anything. So there is, it's almost like cooking. So there's a lot of any good histologist will tell you this. Now, they have their own methods. Yeah, in the sense, there is some degree of replication. There is their own version of the staining, right? Once you do all that, what do we do, right? Generally, in the general procedure is you had a glass slide, you put it on a microscope, I view it, and I do some analysis of it. In this day and age, as I said, we were talking about human brains. You have 7,500 sections, all very big. Somebody manually studying that is going to take eons, not just years. It's beyond their lifetime. So we started imaging, or we started digitizing these entire structures. Okay, and then there is a further pathway that takes us to analyzing, where are the cells, what are the fibers, how is things connected, and so on and so forth. This is kind of how you do it. This is just a, overall picture of, how to do it, kind of thing. Um, So as I just mentioned, for the structural architecture, we basically are looking at these nodes, which are little cells, and we use a special kind of staining. The most commonly used one is called Nissl staining. There are two chemicals that are used. One is called crystal violet, other one is called thionin. I won't get into the details of it. And the second thing that we want to use is to look at connections between these cells. In the brain, particularly, these connections are covered in a, for lack of a better word, in a sheath called myelin. You would have learned in your introductory lecture, or the introductory self tutorial. It's there about myelin. So we actually stain for this myelin, actually, where you can tell, this is a fiber or not. Is that the only way I can study connections? How do I know? Everything is made of myelin, right? So how do I know this area is connected, and how is it connected? Is the input cable that's going, or is an output cable that's going? How do I exactly do know it? The way generally things are done in animal experiments is that we use tracer injections. Traces, there are various types. There are chemicals, there are enzymes, and there are viruses that we use to our advantage because of their features, right? in the mouse brain architecture, the majority of this data is from a genetically modified virus called augmented adenovirus, or AAV, and they have fluorescent proteins stuck to them. So every time the virus infects something, you will actually see it in the imaging thing. So what you do is you basically inject this virus onto a particular brain area, and you have small enough volume so you can pretty much isolate one particular area or one particular sub area within an area or the different layers, and then look for where the connections are, right? There are two ways this is going to happen, right? There is inputs and there is outputs. Okay, so when you inject something, wherever the virus is going to spread basically it's going to infect all the cells, and the output of these cells goes to different areas. We call this anterograde projections, which is basically the output of that particular region. The reverse is also true. We have specialized viruses or toxins which are very specific for they don't infect a cell, but they actually go backwards through the axons that supply that area back to the regions that supply them. So that way we can characterize inputs. This is the way we actually study what is the input to a particular brain area and what is the output to a particular brain area, right? Of course, this is pretty much very easily done in animal models where you can control this. Unfortunately, we can't do this in the human for obvious reasons. Okay, so I'll talk a little bit about what exactly is happening at IIT Madras. So we have embarked on this project where, I just mentioned it's very, very difficult or next to impossible to do this histology or mesoscale resolution analysis of a large human brain, right? That's exactly what we're doing here. Right, because we believe the technology has reached a point where we can push the technology to a particular point so that we can actually generate this possibility. Of course, I just mentioned we can't section, the section is too big. So we are developing very fast high throughput techniques which enables us to do this, because there are a number of sections I just told you it's going to take eons, probably more than a lifetime to do this, but we are kind of accelerating this process by making it high throughput, right? So we have a large body cryotome, which is what we are using to cut very, very large sections. And Professor Partha Mitra actually pioneered a technique called tape transfer. What happens is that instead of, because in the previous days, people used to, cut a block, they used to take it with a little piece of brush and then put it on a glass slide. Okay, that used to have a lot of problems because, obviously you are physically moving something to something else, and the actual loss of tissue is quite large. The tape transfer technique kind of avoids all that, because what happens is you have a tape that goes onto the actual slide. It sticks to the section, and when you make a 20 micron section, the 20 microns section is actually on the tape, and the tape is basically made of a special UV uncured dye. So if you apply UV, a small burst of UV radiation to this tape, the tape basically comes off, right? So it allows for easy transport of the section from where you cut all the way to the glass slide without any significant damage to it. Right? Because remember, our goal is to image the whole human brain in its entire resolution, not pieces, not cuts, not make chunks of it, the whole brain, right? So it's very important to collect all sections and also make sure the sections are not damaged. And these two techniques particularly has allowed us to do that, by cutting, by ensuring that we can cut a number of sections reliably and transfer this onto a glass slide reliably. In addition to this, I just mentioned, right, we have to use certain staining protocol to actually visualize it. If I was doing manually, I would have taken a glass slide, dipped it in some solution, have a series of solutions to get it right, and then cover slipped it. The team at IIT Madras basically is developing automation for this. So after all, it's as I just mentioned, it's like cooking, but it's always better to automate this procedure. There is some degree of a feel, if I want to call it that, or a gut feeling about it, but we can always automate it. So we are currently, and there, obviously the feel factor in a human, we also have to be very consistent, right? So our automation basically results in this. So we have automatic robotic arm stainers, which basically takes the bunch of slides, dips in whatever solutions we need to for X number of time, and also will cover slip it, and at the end results is you already have a stain section that's on a glass slide for further analysis. Okay. It's important to develop these automation simply because you want your procedures to be high throughput. Okay, now comes the bigger problem, right? Which is how on earth are you going to image something that's 15 centimeter by 15 centimeter by 20 micron, right? There is one commercial equipment that is available, the Huron LE120, one in the entire world. But unfortunately, it does the job, but it's extremely slow. Right? So if you want to go something like 0.2 micron per pixel, it's going to take you 2 hours approximately to image just one section, and there are 7,500 of them, right? So IITM is again, leveraging their world-class engineering skills to develop a kind of a fast line scanner, which is capable of scanning at a much faster rate. Our eventual goal, or you want to call it your aspirational goal that we are looking at, is something that can go at 100 times faster than what is commercially available, right? And yeah, we're still working on this, and the project is still ongoing, and we have some prototypes, but yeah, watch this space for further updates. Okay, what happens after we've mounted this? We stain these glass slides. You can see it. It's on a glass slide, which means we can start looking at this data, right? How on earth are you going to analyze all this data, right? We'll have to start with something very basic, right? Which is I have 7,500 sections. How on earth do I align all of them? The first section will be different to a second section, and there are multiple stains. So you're doing for cell data, for the cells you're using missile staining, and for the fibers you're using, it's undergoing a different procedure for the myelin staining. So there might be distortions. How do you account for all that? Okay, and the methodology that we used is called registration. You guys will get a feel for registration when we do our tutorial on Thursday. Basically, you need to align these to the same modality to make a coherent structure. You're putting everything together, right? And also to different modalities, right? There are so many things there, right? There is the cyto architecture I just mentioned. There's myelo architecture that I mentioned. In addition, I could have done an MRI to this human brain before itself, right? So I need to align all of these different modalities and make them as a coherent, uniform volume. Okay, that's the first thing that I want to do, align them. Why do I need to align them? Somebody might ask me, why do you need to do that, right? The reason is simple. If I have two human brains, for example, my brain and your brain, I take one particular coordinate, right, some x, y, z coordinate in my brain, and I say this is visual cortex. How do I know it's the same coordinate is the same your visual cortex, right? You can't say it. Or is it the same visual cortex? Is there a discrepancy? How do I know all this? So I need to A, align all of them, and B, I need to check both my brains onto a reference template, right? In majority of cases is the atlas, or it's like a Google map. If in some of the things they don't exist, which means we'll have to think about innovative ways to compare data between align two different brains, how to align them, and then figure out, is your border of the visual cortex same as my border of the visual cortex, or is there a difference, right? So that's where it gets absolutely critical to do registration. Okay. The second thing you want to do is you want to detect the objects of interest, right? As I said, I can't, as a neurobiologist, I would have just sat under a microscope and looked at these glass slides, 7,500 sections, and started counting all these different objects manually. There are 85 billion neurons. We just mentioned it. It is not possible in my lifetime. Okay. So yes, we are going to be developing automatic algorithm. We are actually developing automated algorithm. We'll talk a little bit more about that. So you want to detect germs as the cells, fibers, etc., etc. Right. Your next lecture by Dr. Shamik Banerjee will actually tell you as to some of the techniques that we use to actually do it. Okay, and the next part is to reconstruct the path for all the different nodes that we just mentioned, right? So we want to, let's say we found a fiber, right, and it's going from point A to point B. Because I've registered everything in a reference space or a template, I can now say this particular node was in visual cortex, this particular node was in auditory cortex, and we think this is what the density of the connection is, or thing. And the third thing, obviously, it's always good to get a reconstructed back and to get a 3D perspective of all my cells and all my connections. Okay. And then eventually, if I have enough samples, then what I generate is the comprehensive map of the brain itself, right? Okay, let's talk about registration. Okay, we just mentioned it. Registration is the process where we need to align our cut tissue. And we also mentioned this. We need to match them to a template. I put the template in quotes because if you don't have an atlas, you don't have a Google map. How are you going to know this, right? Then you just compare, two separate brains. But the whole idea is simple, right? If I have a coordinate, I give you some random coordinate. The numbers there don't mean anything, but they should mean exactly the same for two different brains. Okay. If they don't, then we need to figure out whether it is, it's because the brains are different, or is it because of some other reason, right? That's why we need registration. Okay. What exactly do you use for registration? Right? How do you even register something? Uh, we utilize transformations for this, and you can the way you can think about it is, I can push an image to the left or right. Okay. But remember, when you push something from the left to the right, I am also pulling from the opposite direction, correct? These kind of transforms are called linear transforms. And there are generally four types, right? There is translation, which is basically I move the whole thing left, right, up, down. There is rotation, which is rotating on its axis. So basically, tilting it left, tilting it right, etc., etc. There is zoom. You can zoom in, make it bigger or small. And there is shear, which is pulling from one direction, which means the other side is also going to know. These are all linear transformations, and the GIF that you see there basically shows all these different types of linear transformation that is applied on the brain, right? There is also the non-linear way, right, which is if I pull one thing and the other side doesn't change, then it's a non-linear transformation. Okay, but we utilize all of them to get let's say a test image onto a template image. Okay, that's what you're trying to do here. I just mentioned it. We have multiple modalities. We have the MRI, we have myelin, and we have thing. So which means we need something that can do it in a multimodal way, a lot more complex than a simple. Here is a 2D image. Here is another 2D image. Can I align it? Yes, you can. A simple linear transformation should suffice for that, right? But do the same thing for in 3D. That's going to take some time, right? So you're going to create everything in 3D space. That's going to be complex as well. And now I have multiple modalities, which means I'm going to create something in a dimension that's greater than 3, right? And this is called multimodal registration. We have been working on this problem. Obviously, this problem hasn't been solved yet, but in case you're interested, we are actually working on this problem to actually get it, um, you know, uh, to have a reference point and then get a transform coordinate, and then align it to an MRI, and then align it to a different technique called the example that you see here is from a mouse friend from Mitra Lab. There are several different categories here, right? So there is the MRIs, there is NISL, and there is something called a serial tomography, STPT. And then there is an atlas itself. The mouse brain is well studied, so it acts its own atlas. And there is not big variations in the atlas. So you can actually use a reference template as the atlas, right? And here is just a registration technique that we have been developing. Which basically allows us to do all these registrations, right? This is developed by Associate Professor Daniel Twart and Professor Michael Miller. Daniel used to be in Johns Hopkins, but with Mike, now Daniel is a faculty at UCLA. Yes, we can do the same thing for the human brain as well. So here is just an example of what you see there, and the two stains that you see where you're showing the human hippocampus. The left hand side is the myelin stain, which basically tells you all the fibers that have been stained for myelin. If you zoom in at a very, very small resolution, you kind of notice that there are these small wriggly wires, and you can see that the density of these wiring is very different, right? All the different regions are different. And of course, the right hand side is your NISL staining, which basically is staining your cells or nodes. And if I register the both of them together, I can kind of say, here are my cells and here are all the fibers, and we think this goes from here, right? That's kind of the picture of why you need registration. In addition to it, like obviously, we just mentioned about traces. So we've injected something, and then we are, looking at where exactly do these cells go, or where do they output? Can I literally look at it and go on the left hand side? You see there is a big bright spot there. That is basically a fluorescent injection, right? And you kind of make out, at least for the red ones, you kind of make out the red is going to different areas. You actually see the spread going from that injection outwards. And you want to detect this. You want to detect this signal, right? If you were manually doing it, you would have just gone and said, oh yeah, there is this much in here, this much in here, this much in here. We are actually generating at IIT Madras and with CHSL, we are generating automation to take care of this problem. Now, machines are brilliant at pattern detection compared to us, which means we can actually utilize their capabilities to actually get this kind of automatic detection going. Of course, we'll talk a lot more when you come to your tutorial. You'll realize that obviously this is not absolutely perfect. You do need a little bit of human in the loop, but it's several orders better than somebody manually marking these, rather than me going in and just proofreading it and getting away with it, right? So we are also implementing, the segmentation mechanisms, automated segmentation mechanisms, which kind of tells us this is a cell, this is a fiber, this is a, those kind of differences. And we are using something just not simple ML or RCNN. We are also basing it on a very solid, theoretical basis, which is the discrete Morse theory, and it seems to work well. But and I won't get into great details about this because in the next talk you will hear Shamik talk all about this. Okay, you've done all this. So what exactly do you need to do next? Okay, so we want to create a Google map of the brain. You need to find out where all the different regions are and where does this road go to, right? And we have been working with Lucas Maggi, who's a grad student, and Professor UC Wong from UC San Diego, to so we have a the rough picture is you have one section which has some detections. It's telling you that there is a connection from here, and you have a next section which is obviously is a 20 microns away, but there something else is starting from somewhere else, right? When you put everything together in 3D, there's going to be a gap because it's 20 microns thick, so we can't see everything in that 20 micron thick, right? And there is multimodal. I told you there is a NISL and there is myelin, and there is NISL, there is myelin. If it's fluorescence, there is also a third structure which is fluorescence, right? So between, sometimes you can go 20 microns to the next one is 80 microns away, correct? Which means I cannot connect these segments that I've detected. We basically borrow, it's exactly same as Google Map. Google Map is never going to map every single centimeter of your road, right? So they map, they know there is a road here, they know there is a road there, and they assume that it's connected. We do exactly the same thing. Again, we're going to base it on discrete Morse theory, and we've been working on how to connect these different segments and to create a skeletonization, we call it a skeleton of these connections. Basically developing the Google map of the brain, right? We are working on these procedures right now at IIT Madras. So anybody who's interested, please feel free to call us. And that kind of concludes my part of the talk, which basically introduced you to how we do it, and this is what we do it. Dr. Banerjee will be talking in the next lecture about more specific as to how to use machine learning and all these different techniques to do these kind of procedures. All right. Thank you.